Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate datasets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here today with Dr. David Bumcrot, who's the Chief Scientific Officer at CAMP4 Therapeutics. David's career has taken him through the heart of two of the most revolutionary breakthroughs in biotech, RNA interference and CRISPR gene editing. And I think we're going to talk about a third today, which is pretty interesting, especially to me. I'll talk through my background, why I'm so excited about this. But David was one of the first employees at both Alnylam and then later Editas Medicine, helped shape those Nobel Prize winning science discoveries into translational platforms to benefit patients. And he's at CAMP4 today, where they're focusing, as I mentioned before, on something called regulatory RNAs. And this is particularly close to my heart, having spent a lot of my career working on haploinsufficient developmental disorders and other rare diseases where the therapeutic options are extremely limited until relatively recently, thanks to the work that you and a number of scientists around the world are doing. So David, thank you so much for taking the time to be with us today.
Thank you. It's a pleasure to be here.
I would love to start with RNAi and CRISPR and some of your background in some of these transformative companies that people know. I'd love if you could just talk a little bit about your experience there, what that taught you, how that shaped you, and what you bring to your career today through those experiences.
Yes, of course. So what's fascinating, I think, about those two technologies is that they were really both the revolutions of their time. So, when RNAi came along, that was really considered a revolution that we could apply to human health. And then after that, CRISPR came, and they were both very heavily covered in the popular media, in the press, had a lot to say about them. They were both based on mechanisms that are sort of natural mechanisms that occur in organisms, but they were first discovered in sort of lower organisms, not in humans or mammals. And then it was discovered that they could be applied to develop drugs. As you mentioned, they were both Nobel Prize winning discoveries. And so, therefore, it was really kind of the responsibility of the companies, Alnylam and then Editas, respectively, to sort of take these brilliant discoveries and sort of not mess it up, to figure out a way to make these actually into drugs. And that was a long process. And there were a lot of things that were in common between them that really, I think, helped me because I really enjoy working at companies that have new platforms. And that's why I find CAMP4 also so satisfying to work for. And, a few recurring themes. One was that, you really have to, as you're optimizing the platform, figuring out how it's going to make drugs, you also have to start drug development programs. You really can't wait until everything is perfect with the technology you need, because there's things you're going to learn only when you actually start a drug development program and figure out, what are the challenges? How do you make these molecules into things that can act like drugs? What will regulators need to see? How do you measure safety? What does safety even mean? And so those are, I think, some of the key themes that were common between those two.
Are there any stories behind either of those where you maybe waited too long, or you learned something really unexpected as you started to bring it from this, pure platform concept into let's get it into the real world and really understand how it works in patients and learn some of these important lessons?
Yes, I think for RNA interference, really appreciate the importance of chemistry, I think took a while. The first thought was, well, let's keep these molecules as simple as we can keep them, as minimal chemistry as we can. But it turned out that these kinds of small bits of RNA had some properties that needed to be engineered out of them.
Oh, interesting.
They would induce a type of an immune response, the so-called innate immune response. You could have real issues with these as drugs, and those were solved with chemistry. And I do have to give a lot of credit to people like Ionis, which used to be ISIS, now Ionis. They did a lot of work on chemistry with single-stranded antisense oligonucleotides. A lot of that was also applicable to RNAi. And so together, the companies, really through decades of work, discovered what it was, what it took to make something like a double-stranded siRNA, which is very non-drug-like, into a drug-like molecule. And I think we were all surprised at how long that kind of took. And as it relates to RNA interference, I think things like, well, what does it even mean to measure safety with this type of technology, and to really spend a lot of time figuring out, how can we measure off-target effects? Where is editing occurring that maybe you didn't want it to occur? And things had to be developed. And then some of the founders of the company, like Keith Jung and others, came up with technologies to do that. I think we underestimated how long and how difficult that would be. So those were some surprises, but the company solved both of those issues.
I listened to a very interesting podcast with Stan Crook from Ionis, and he was talking about one of the big advantages of RNA is that you have this compounding where when you figure out this chemistry, it actually applies really broadly across lots of different therapy areas and modalities, where gene editing, you almost need to solve these problems in a different locus or a different organ or a different delivery method. You might have different off-target effects, and so there's not that level of scalability. I'm curious to get your take on that, whether you think about it similarly or see it differently.
Yeah, I think so. Stan is absolutely right about the broad applicability of chemical modifications to different kinds of RNA-based drugs, whether they're single-stranded antisense oligos or double-stranded siRNAs. It turned out that the way you could solve these problems, like induction of the innate immune response or improving stability in blood and in tissues, could be applied across all those platforms. So you kind of had a sort of a stereotypical pattern of chemical modifications that you would put on them, and it would solve those problems. And I think the point that he's making about genome editing is really that the way you get those components that you need into cells, there's lots of different ways to do that. It can be with a gene therapy vector. It could be with messenger RNA encoding, CRISPR. It could be with the CRISPR protein. And to decide which of those is most applicable to the tissue you're going after really kind of makes each one its own real kind of project. And also the whole idea of in vivo editing in tissues versus in vitro in cells that you would then put into patients, really a lot more variables of that type for those technologies.
We have historically different technology waves. We have biologic companies that have gone from zero to major scale, Regeneron and Genentech and Amgen and so on. We have the same, actually, I think, in the RNA wave with Ionis and Alnylam in particular. We haven't really seen that in the gene editing or gene therapy wave. I'm curious to get your perspective on whether there's something fundamentally different there, that we're never going to see that, or is it a matter of timing, and those kind of full stack companies focused in those therapy areas or those modalities are going to emerge at some point?
I think they will emerge. I think as the different technologies keep to evolve. So, the next generation types of genome editing, different kinds of base editing, different types of sort of gene writing, ways to insert genes in different ways. I think that a lot of companies are somehow feel a bit more comfortable with some of those technologies. They can see that those can be controlled better, that they can maybe be more specific. I think that people were really, a little bit scared of CRISPR when it first came out. And then when Chinese scientists edited human babies, this was kind of a wake-up call also, that there's things about this technology that maybe we better wrap our minds around. But now that companies are doing actual in vivo editing in human liver, and seeing positive results, I think that this is giving a comfort level, and I think we will see some of the big players stepping in.
Yeah, I'm interested to see how that all plays out. I want to talk a little bit about CAMP4's approach. As I mentioned in the intro, this is something that immediately caught my eye when we got in touch, because I'm really excited about what you're doing, in particular for the rare haploinsufficient gene. The disease is caused by haploinsufficient genes. Could you talk a little bit about the regulatory RNA concept that the technology is based on, and just give people a sense of what you're working on, because I think it's really cool.
Yeah. So the RNAs that our drugs target we call them regulatory RNAs, and they're part of a broader class of RNAs that have been known about for a while called non-coding RNAs that people have been studying for some time. But regulatory RNAs, where they specifically come from is from the regions of the genome that control gene expression. So the way genes are controlled is by these elements called promoters, where the messenger RNA starts, other DNA elements that are called enhancers that operate at a distance, and these control how genes are expressed by binding transcription factors. And it turns out that these elements themselves, the promoters and the enhancers, are also transcribed. RNA is being made off of them. And this was actually known. This has been known since like the 2010s. But these RNAs were completely ignored. They were thought to just be an artifact. There's some RNA being made. It doesn't really do anything. And it really wasn't until much more recently, and I'd say much of this work comes from one of CAMP4's scientific co-founders, Rick Young at the Whitehead at MIT, that these RNAs actually play a critical role in controlling the level of gene expression of every gene in the genome. So if a gene is being expressed in any cell type, there will be these regulatory RNAs associated with it. And what they do is they sort of tune gene expression in response to the environment. They control gene expression within what I would call a physiological range. So they control gene expression plus or minus, 2 or 3-fold, let's say. And this is ideal for haploinsufficient diseases, as you mentioned, because by definition, those diseases are caused by having half as much protein. So from the wild type gene, if you can increase expression modestly, you can have a real impact. So what did we discover at CAMP4? We discovered that small pieces of modified RNA, antisense oligonucleotides, maybe 20, 23 nucleotides long, can be designed to bind regulatory RNAs and actually give an increase in gene expression. You can also get a decrease, but we're completely focused on the increase. We think that's what's unique about us. That's what's really missing in the field. If you want to knock down a gene, you can use antisense oligos or siRNA, but increasing gene expression. And the way it works is because Rick Young discovered that essentially every one of these proteins that's involved in gene expression, transcription factors and related proteins, also bind to RNA. And so what regulatory RNAs do is they basically form like a kinetic cloud or a trap of RNA around these enhancers and promoter elements when they come together and form what's now being called a condensate. It's where the proteins come together, RNA is there, and determine, how efficiently these proteins will bind to their sites on DNA. And what our drugs do is they bind the regulatory RNAs, kind of, perturb their structure a little bit, alter the balance of binding of proteins, and then the net effect is an increase in expression of the gene that's being controlled by those regulatory RNAs. And it's a universal mechanism, so it can be applied really to any gene.
Interesting. So how easy is it to predict what perturbing a particular regulatory RNA will do to a particular gene's expression? Is that, is it deterministic? I'm sure it's deterministic, but is it clear in the sense that some of them are inhibitory and some of them are promoting, and so you either have a knockdown or knock up, or is it context dependent? I'm just curious to learn about that. You make a change here and what happens downstream, and how you map that landscape.
Yeah, this is very much an evolving space, and CAMP4 is working really hard to understand this. So what I can tell you that what we understand very well, what our platform was really optimized to do, is for a given gene in a cell type that we're seeking to upregulate, our platform has allowed us to build a model that will tell us, here is where the candidate regulatory RNAs will be made that will very specifically control that gene. Whether it's up or down, this is where you want to look. These are where the reg RNAs will come from that will control that gene and no other gene. And then we did a lot more work to actually get the full sequences of the regulatory RNAs. But that's also really important because these RNAs, they're not very well annotated. You don't find them in the public consortia. But now to get to your question about, what's the actual function? What if you were to engage or bind a reg RNA? That is still somewhat empirical. And what's really interesting is it turns out that, since most of our regulatory RNA binding drugs, the ASOs, they don't degrade the regulatory RNA. They act in what we call a steric manner. They bind and alter its structure. And it actually matters where on a particular reg RNA you bind. In some areas, you can get a decrease in expression. You move over, you get an increase. And it's because of the suite of proteins whose binding you are perturbing could have a net negative or a net positive effect. And it's a little bit different for each gene. And we can tease apart those mechanisms. So I think, any given regRNA could have both a positive and a negative function depending on how you perturb it.
Fascinating. There's an interesting topic to go into here, which I've had with a lot of guests on this podcast about the role that genetic targets play in either biotech or large pharma's strategy, and it's very hard to compete with someone like Regeneron on coding, exome sequencing and finding rare loss of function variants. But this is really interesting because it's a space that very few people are focused on, right? And so you don't have to reinvent the wheel in terms of RNA, the modality to actually get in and have this effect through the steric binding. But there's really a lot of opportunity to find some novel biology through understanding the input and output from perturbing these regulatory RNAs, right? Is that the general concept?
That is exactly right. I think it really is an advantage that the actual drugs that CAMP4 is developing are chemistries that are sort of, it's a well-established modality.
Yeah.
They look very much chemically. They look like splice modulating ASOs. They look like oligos that different companies are already making that are already approved drugs. The chemistries in our ASOs, they're available to us. They can be manufactured at scale.
Yeah.
Regulators are comfortable with them, used to seeing them. The novel part is the actual targets we're going after.
Yeah, that's super interesting.
Yeah.
So how you alluded to this problem, and we discussed it earlier, you have a platform where there are thousands of haploinsufficient developmental disorder genes. I know that because I spent years of my life staring at them.
Yeah.
How do you think about how to break that problem? Now we have a platform, you can go so many directions, but you need to do what you said and start to see what happens in the real world.
Yeah, there's a few different sort of layers of kind of filters we go through, and really one of the first is where have these types of ASO drugs been successfully delivered in human beings, and this is growing, but the sort of the most successfully approached tissues so far, one has been hepatocytes in the liver, and a second is, and this is growing, is in the CNS. Mm-hmm. Now, as it turns out, there are a lot of haploinsufficient diseases in the CNS. So this is really a very fertile ground for us to go after. So, we were initially focusing on those two tissues. We're really veering more into the CNS, and our future programs will likely mostly be in the CNS. Another thing that's interesting is, for haploinsufficient diseases, it makes great sense because of the amplitude you need to increase the gene. It's within a range that we can achieve. What's also true is that a lot of these haploinsufficient diseases, there's genetic evidence that you likely don't have to get expression all the way back to 100%. And we know that because of genetic evidence. Often in these diseases, for the one mutated allele, there's like a genotype-phenotype correlation. There's an allelic series in humans where some mutations cause more disease, some cause less, and it tells you that maybe only getting halfway back would be enough. And that's really important because the kind of effects that we get in vivo tend to be sort of maxing out at around twofold or maybe a little higher. So it's really important for us that you are confident, that you have real conviction that the kind of increase we're getting will have an impact on disease. So, those are some of the key features.
How do you connect the dots between what that, and I think we may have defined earlier, but for those who aren't as familiar, haploinsufficient genes, if you lose one copy due to a loss of function mutation, for example, there are many genes in the genome where not having enough expression from the one copy results in some kind of damaging phenotype. And like you said, you may only need 60% or 70% of the dose to be phenotype free, but if you drop down to 50, and there's often some buffering, right, where the gene starts to work a little extra hard, so maybe the one copy gets to 60% on its own, but there's some threshold that you need to cross.
Yeah.
How well do we understand what that threshold is for all the genes that you're targeting? Because that is probably a big unknown as well to some extent.
Yeah, I think there's different levels of evidence supporting that for different diseases. And for some, you will go into the program knowing that you're going to carry a little bit of risk, that there's maybe some controversy in the field, or it's not entirely sure whether a very small increase will help. The two types of evidence, one, I think human genetics is among the best evidence, where, like, for example, if the gene is an enzyme or something whose activity can be readily measured, there are model systems where sometimes you can figure out where's the threshold you need to cross. For others, it's maybe based more on studies done in model organisms in mice, for example, where you don't know for sure it translates, but at least you feel pretty confident that it can. So it's really, and then talking to as many experts as we can, really understanding the field, we build that conviction. Sometimes there are even drugs that are maybe imperfect.
Right.
There's issues why they haven't been taken up very well, but they at least give you some confidence that if you do engage this gene, that it can have an effect. So those are the kinds of evidences that I think we bring to bear.
What do you think about other approaches to solve this problem? Like I think a lot of this is way earlier in preclinical than what you're working on, but I've seen, proof of concept of using CRISPR fused to P300, for example, to drive the promoter of the one healthy copy of the gene. Is there really any other like clinical stage technology like this, or is this the first shot that we have at addressing this class? We need to upregulate rather than downregulate.
Yeah, so like all of the sort of the modified versions of nuclease dead Cas9, so epigenetic
Yeah.
upregulators, things of that nature, doing things like expressing different tRNA variants that can then suppress nonsense mutations, things like of this nature.
Oh,
So
right. Yeah, interesting.
it's all interesting. What all of those approaches have in common, I think, is that the delivery challenge, they either have to be delivered as gene therapy
Mhm.
vectors, like AAV or big, long, complex messenger RNAs. They're not really like drug-like, whereas ASOs are extremely drug-like. But I think another technology that probably is analogous are the splice modulating ASOs. For example, Stoke Therapeutics is seeking, they do upregulation with a splice modulator, and they're in phase 3 for an epilepsy disease, Dravet syndrome. And so they're solving upregulation in a different way. Their target
Mhm.
is the messenger RNA. We think that this is a very exciting approach, and it's actually paving the way, I think, for these types of drugs, which are similar to ours. Our mechanism is different. Ours is upstream of theirs. It's transcriptional. We know that our mechanism is applicable to every gene in the genome. I think that the splice switching is broadly applicable, but probably not as broadly as ours. There aren't there's not enough of this misspliced messenger RNA for every gene you want to go after. There definitely is for Dravet, but probably not for every gene. So I think that's probably the closest comparator is what
Yeah.
Stoke is doing.
Yeah, interesting. You have a few things on your pipeline. You're working on urea cycle disorders. I think you have a program in SYNGAP1, which is developmental disorder, and also you have common disease applications like GBA1. I'd love to get a sense of the timelines for those kind of programs. When do you expect to start treating the first people in early clinical trials, and how do you think about how this plays out from here?
Yep. So, the program that is now in a phase 1 trial, but it's in normal healthy volunteers, is the urea cycle disorders program. So that's our liver program. It seeks to upregulate an enzyme in the urea cycle called CPS1, which itself is very rarely mutated. So it's a little bit different. So that one is not going after a haploinsufficiency per se. That one is more of an augmentation approach, where it turns out that if you elevate expression of CPS1, you can overcome mutations that are downstream in the urea cycle and restore the normal metabolism of
Interesting.
amino acids and keep ammonia down. And we presented data on that in a mouse model. So that one, we have plans to look into going into a subset of patients, which are actually who have a mutation in a different gene called OTC, in the coming year or so. And we're looking into that as part of that phase 1 trial. But I think we're really excited about moving into the CNS. And so our most advanced program there is for the SYNGAP related disorders. That is a bona fide haploinsufficiency. Definitely is caused by loss of one of the 2 genes that encode SYNGAP. There is evidence that you probably don't need to get expression in neurons all the way back to the wild type level, so it fits beautifully. And for that one, we just last week presented some very compelling data at the ASGCT meeting, showing evidence that we can elevate SYNGAP enough in a mouse model to correct many of the behavioral deficits in that disease. Also show that our drug candidate works in non-human primates. And so that one is on track to enter sort of the pivotal tox studies that would enable entry to the clinic. We're going to do those studies this year, so that could be in the clinic next year.
Amazing.
And then behind that, I would say, if you think about, the bigger diseases, where could this be applied, like Parkinson's disease and so on? That's sort of the next wave. For a company of our size, obviously we would need to partner a program like that to really go into the clinic in a large disease. We feel we can take the other ones pretty far ourselves. So, I think applying this technology to bigger diseases will be the wave that comes after that.
And GBA1 and Parkinson's is a loss of function case, right? That's like SYNGAP1, one of these haploinsufficient.
That's right. Yeah.
It's interesting as well to think about jumping from such a set of rare populations as well to a common population, how you think about designing a clinical development plan and all those kind of points, right? Because you have an entirely different set of challenges to solve with each of those different worlds.
Yeah, that's true.
What have you learned about clinical development and taking early, preclinical new technologies into the clinic? What are some lessons that you learned along the way on how to make good decisions and do things early that set you up for success in the future, rather than paint yourself into a corner by accident based on some of the decisions you make early?
Yeah, I think we did learn it at Alnylam and Editas to try to communicate with regulators fairly early in your program before you've committed fully to what you're going to do, to start having a dialogue, start understanding what will the expectations be in terms of what they want to see for safety, for specificity, before you commit to designing your full tox program. I think that, really taking the lead and educating into, like, for Alnylam's case, so FDA, about what the technology is. The FDA would say, you guys are the experts. Tell us, help us understand how are they similar to other kinds of drugs? Why are they? How are they different? Help us. We'll work together to figure out, what do you need to show that's a bit different about these? And I think jumping all the way to CAMP4, what we've learned from talking to regulators is, they're very comfortable with antisense oligonucleotides, the chemistry, the sort of the class type of issues to keep an eye out for. But they just wanted us to teach them about the targets. What happens to your regRNA target? Like, how long does the effect last? How can you assess that? Make us comfortable that kind of what you're doing, but what are you perturbing in the system and for how long? And they've been very open to these kinds of discussions about using a combination of in vitro and in vivo methods to do that. And so if you engage with them and have these conversations, I think that's really, really important.
That has me thinking about a question of how you actually discover these regulatory RNAs in the first place. You mentioned that this has been overlooked for a long time. When I did my PhD, I was focused on the non-coding genome and developmental disorders, actually, and looking at ultra-conserved elements and these other parts of the regulatory genome that we had a hypothesis maybe were involved, in the sense that if you knocked one of them out, they could perturb gene expression enough that you'd cause a phenotype. But I remember learning about the regulatory RNAs at the time, and some people saying it's just kind of garbage coming off the transcriptional machinery. But as you mentioned, that changed. How well is that mapped through ENCODE and some of these big consortiums versus stuff you may need to do internally, where you're doing your own mapping because you don't have the right tissue time point? This is a big matrix to fill out, right, of tissue, time point, regulatory moment in the organism. So it's a hard problem to solve.
Yeah, so I think a lot of those consortia, they're extremely high quality. They're very good. A lot of the data in those consortia come from established cell lines. A lot of them are tumor cell lines, cells that have been in culture for a long time. There's less data in actual primary human cells. And that's really important. And that's what CAMP4, I think, is really a leader in. So, we've done this, what we call mapping, the transcriptional mapping, where we use a whole bunch of next-gen sequencing assays to map out not just every gene that's expressed, but every single one of the active regulatory elements, every enhancer, every promoter, which enhancers and promoters are interacting with each other. We have all those data from about a dozen different human cell types, and we make it as close to the primary human cells as possible. So we can source primary human hepatocytes, for example, from different suppliers.
Yeah.
As in the CNS, we mostly use cells that are differentiated from induced pluripotent stem cells, from iPSCs, because it's really hard to get primary neurons. And so we have, I think, the most relevant data set you'll find out there of exactly how are genes being controlled in cells that are closest to the human tissue. And a lot of that information is not available. So for a given gene, whether it's SYNGAP or the genes that we're modulating in the urea cycle, I think we have a much better picture of how that gene is actually controlled in the neuron or the hepatocyte, respectively. Then the second thing is the actual RNAs, their full sequences of these regular RNAs. You find very few of those in the public databases. And the reason is these regular RNAs, they're often pretty short-lived. They're being made off of these elements, and they tend to they turn over pretty rapidly. In fact, they're likely functioning while they are being transcribed and are still sort of tethered to the DNA. And so when our ASO is there, it's sort of interacting with these as they're being made. So they kind of fall under the radar in a lot of these large-scale RNA sequencing initiatives that are out there. Whereas we have catalogs of tens of thousands of these sequences from different cell types.
Yeah, that's fascinating. I feel like there's a whole world to explore there that you mostly aren't looking because the science is only just catching up to the biological relevance of these elements, right?
Yeah, that's true.
In SYNGAP1, is there a treatment window that you're thinking about? Because for some developmental disorders, I think it's understood that it's generally better to treat as early as possible, but some have a surprisingly long therapeutic window, right? Where you can go into late childhood in some cases and see an impact. I'm wondering what you know about that, but more generally about the class of diseases you're trying to treat, because that has a pretty big impact on how many people you can help and what you can do.
Yeah, so I think in general for these sort of developmental epileptic encephalopathies, the earlier you can treat, the better, because these children are missing developmental milestones, and so ideally you do want to get as young as you can. But you're right about this sort of prolonged treatment window, and indeed, and some of this evidence again comes from some pretty clever mouse models where you can genetically reintroduce SYNGAP at different times in the life of the mouse. And what's been published is that even into adult mice, if you wait until they are adults, if you reintroduce full SYNGAP expression, you can overcome a variety of the phenotypes, including the seizure phenotype. So while early intervention will be better, don't forget the older patients. And SYNGAP, we know that our drug should be able to help them as well. And that's really important because we'll likely have to start in slightly older patients and then make your way into the younger ones as the drug development proceeds.
Yeah, and that is driven by FDA being conservative about safety and that sort of angle, right? Is that usually
Yeah. Mm-hmm.
Yeah. As we broaden out a little bit, I'm curious to get your perspective. If we look beyond what you're doing at CAMP4 and beyond RNA therapies, maybe in general, what are the other technological innovations you're excited about? What do you look and say 5-10 years? I think many more people are going to be talking about or seeing impact from particular area.
Yeah, I have a slightly different response to that, I think, than you might expect. So on the one hand, there's a lot of great developments, obviously, as different proteins are being engineered, whether it's even the next generation of genome editing or gene insertion, technological advances in being able to analyze the whole genomes of single cells. That's all terrific stuff. But the thing that kind of excites me the most is it's not really a technology, it's more of a framework, which was, I think, most recently exemplified by what we heard at ASGCT last week from the group at Penn and Harvard, and to be able to kind of make a bespoke therapy, a genetic medicine. In this case, by the way, it was the urea cycle disorder. This right. Young boy had a mutation in CPS1, which is actually the enzyme that we're upregulating. Yep. And what the group at Penn and the others working together were able to come up with is a way to develop essentially a bespoke therapy in a very short period of time. And a lot of that involved, not just collaboration among different institutions, but also with companies and with regulators. And I believe that, and by the way, another example, a little while ago from Tim Yu was able to working with the people from Ionis and with Stan Crook's initiative to make a bespoke splice modulating ASO for a young girl that had Batten's disease. So these kinds of examples, making these frameworks, I think will have ramifications for the more standard types of drug development. I do believe that they're showing that there are certain things that can be accelerated, that there's ways that you can maybe test these in human beings, safely, but more rapidly than you have before by, now that we understand, but by then really sort of, pushing on the gas pedal so quickly that there's ways to answer critical questions about safety probably more quickly that will allow you to get into the clinic with potentially therapeutic doses sooner. And that, to me, I think is going to be transformative for a lot of these genetic medicines.
Interesting. I totally agree with you. And I think between Lorem and what Stan Crook's doing and that example you shared, I had the CEO of Everyone Medicines on recently, who's taking a similar approach. I'm really excited about that. This point you make about the quicker read on safety. What's the approach? I'm a little bit ignorant around how this. I always assume that because the unmet need is so high and the urgency is there, that it's just a different risk tolerance. But are there ways to actually get that safety signal in vitro or in some way faster to actually get that signal rather than just saying we're gonna take a risk-based approach on this?
Right. So you're absolutely right that a key factor of that was that essentially this was, a life or death period of time. You're absolutely right about that. However, I think what it has forced a lot of us to do is to try and, to show that some of these in vitro to in vivo tox assays actually do translate. So there are things that, as long as you do the experiments in the right cells and in the most best characterized, let's say iPSC differentiated neurons, you can make discoveries that have been shown to translate into primates and into humans. And I think agencies are getting a little bit more comfortable with that. Now, you're still going to, yes, you do have to do the tox and look at recovery, but I think the net result may be to allow us to go in with our initial doses that will be closer to what we really have confidence will be therapeutic. You won't have to go in such low doses, particularly in the US for oligo drugs in the CNS, very conservative. It's really difficult. And I think Stoke had to live through this. That's part of why they went overseas and were able to increase dose levels a bit more rapidly. I think that, just getting comfort with what these drugs have in common, things that you don't have to that have already been solved, that you're pretty confident will allow us to advance our doses more rapidly.
Yeah, that's really interesting. And I do remember Kent Rogers from Everyone Medicines talking about this a little bit. Can we make the case with regulators that there's a core safety package that underpins doing this on a one-by-one basis that allows us to do it a little more quickly? I love this, and I think sometimes we have a ton of technology in front of us, right? Between gene editing and gene therapies and RNA, and a lot of what's standing in the way feels like it's the way we've always done things, and how do we rethink the system to accommodate these new technologies and what we can do now. On the point about thinking about trials globally rather than focusing in on the US, are there parts of the world where you felt like maybe it's in addition or instead of starting in the US, we might focus in some of these countries because it's a more favorable regulatory environment, or they have a history of engaging with this kind of technology before?
Yeah, I mean, I think Alnylam has kind of led the way there. They're starting most of their programs, I think, overseas. I don't know exactly if it's in Europe or in the UK. Our phase 1 trial that we're doing in urea cycle disorders, we're actually doing in Australia.
Yeah.
And kind of coincidentally, it turns out there's some really high-level experts and centers of excellence around SYNGAP in Australia as well. So we may go there. They have these clinical centers that understand these types of drugs. So I think that's a jurisdiction that I think is looking very appealing. So yeah, a variety of places, I think.
Yeah, amazing. Well, as we wrap up here, is there anything that we haven't covered that you think would be useful for the audience to hear from you? Any takeaways from your career, or things that people can be helpful with if you're looking for, particular hiring anybody or looking for help with something, then feel free to call it out.
Yeah, I think one thing, if I could, just get on the soapbox a little bit, I think as we go back to, these core technologies, whether it's RNAi, CRISPR, I think it's so obvious how important it was that they came out of real basic research that was being done all around the world, but a lot of it in the US, all of it funded by NIH, and just how short-sighted it would be to not realize the benefits that can be reaped from looking at something that at the beginning might look a little bit obscure, but in fact then turned out to launch whole fields, the areas of drug discovery. And so, let's make sure we don't backslide there. I think that would be really bad for our country and the world. I think biotech right now is in a very difficult economic period, and yet I think that there are there are still investors that are looking to put capital into cool and interesting ideas. So, keep the faith. We keep working, and there are still people that are willing to take risks. So companies are being founded. So, don't give up on this field. It's still going strong.
That's right. It is. I think that's right. The science is more amazing than it's ever been, and there are huge commercial and funding challenges, but it is a good time for investors to invest in some very amazing science at lower prices than have ever been possible before, right? So now is a good opportunity to fund interesting biotech companies.
Exactly. Mm-hmm.
Well, David, thank you. This was incredibly enlightening, and I appreciate you sharing all of your experience and the technology you're working on at CAMP4 with everybody.
It was my pleasure. Thank you.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.